Hi, and welcome back to Introduction to Biology for Majors, or BIOL 1224. This is your Chapter 12 PowerPoint from Module 3 of this course. This is from the Openstax book Biology 2E. It's Chapter 12, and it's about Mendel's experiments and heredity. So we're starting off with 3 objectives. We want to be able to recognize the role of genes and genetics for inheritance patterns. We want to be able to apply principles of inheritance in a monohybrid cross with application of genotype and phenotype percentages, and to identify 3 patterns of inheritance in human disease. So on the left of this slide, we have an image of Gregor Mendel. He actually is the father of modern genetics, and we're going to kind of talk about what he did and how he made the discoveries that he made. So genetics explains inheritance patterns. Genetics is the study of heredity, or basically the study of passing down genes to new generations. Mendel set the framework for genetics long before chromosomes or genes had ever been identified, at a time when meiosis was not even well understood. Mendel conducted methodical experiments to reveal the fundamental principles of heredity. We now know that genes carried on chromosomes are the basic functional units of heredity, with the capability to be replicated, expressed, or mutated within our bodies. And genes are a portion of DNA that encodes a specific protein. So let's talk about the basic laws of inheritance. So Gregor Mendel actually used pea plants to study heredity, which was a good choice because it was easy for him to control which plants mated with one another. So Gregor Mendel actually used a hand-pollinating technique, and he did this to the plants, which allowed him to control plant breeding in the experiments that he was doing. Self-fertilizing and cross-fertilizing in different combinations actually allowed him to deduce the principles of inheritance. So if you look over on the right side of the slide, I have an image that kind of depicts what he did and what he was looking for. So he cross-pollinated or hand-pollinated different pea plants to make a tall and a short plant. He was also able to then go into smaller detail within the plants by cross-pollinating. So he had plump versus wrinkled seed pods, round versus wrinkled peas themselves, green versus yellow peas, green versus yellow pods, and white versus purple flowers. So he was able to kind of pull out all of those changes within the pea plants by hand pollinating them. And then that was able to prove that there was some kind of genetic inheritance happening within the plants. So let's talk about dominant and recessive alleles. Dominant alleles will exert their effects wherever they're present. So if a gene is passed down from parent generation to the first generation of new organism, the dominant allele that is present is going to always pass down. So for example, what Gregor Mendel did was he would cross what we call a yellow seed plant with a green seed plant. So one of the pea plants had a yellow seed, which we'll indicate right here. One of them had a green seed. He then self-pollinated all of those plants, and the yellow seed always creates yellow offspring. So the yellow seed color is therefore dominant. So if you look at this image, we have a yellow seed and a green seed that are the parent generations. So he hand-pollinated these, and all of the first generation following are yellow, which means that this dominant allele passed down through the first generation. So the recessive allele, which would then be the green, is masked if a dominant allele is present. So whenever we mask with the dominant allele, this recessive allele does not pass down. So let's talk about some vocabulary words that I want you to know for the next few slides when we're talking about genetics. So a genotype would be our first vocabulary word, and it represents an individual's 2 alleles for one gene. So a genotype then confers phenotype, or a genotype sort of creates phenotype. So basically in my notes, I have it written that a genotype describes genes or alleles. So in this case, I'm using T's as our example of our alleles. So it basically is going to represent an individual's 2 alleles. So if we have 2 capitals, that wants to represent a dominant allele and another dominant allele. If we have a capital and then a lowercase, we want to represent a dominant and then a recessive. And if we have 2 lowercase, we want to represent a recessive. So basically a genotype is going to represent which type of alleles an individual is going to be passing down. And then a phenotype is the description of the appearance and is observable. So it's what you actually see when, for example, running the experiment. So for Gregor Mendel's experiment, he saw tall and he saw short. So phenotype is what something looks like. Genotype is the genes and alleles found within. So then let's talk about homozygous dominant individuals. So like I said up here, 2 capital T's like this will represent a homozygous dominant individual. So if you break this down, homozygous, homo means 1, so we have the same thing. Homozygous means 2 of the same alleles, which in this case are both dominant, which means that they're both capital T's. And these will represent the dominant alleles for the gene. Then we have something called heterozygous individuals. And hetero does not mean 1, it means more than 1. So hetero individuals will have 1 dominant allele and 1 recessive allele. So in this case, it would be 1 dominant T and 1 recessive T. And then we have homozygous recessive individuals, which would be the exact opposite of homozygous dominant individuals. So recessive individuals will have 2 recessive alleles. So just like this, we have 2 lowercase t's here that represent 2 recessive alleles. So these are not strong alleles, they won't be passed down. These dominants will be passed down, and anything heterozygous we'll kind of talk about in subsequent slides. So once again, a genotype represents an individual's alleles, so what's inside. Phenotype represents the description of appearance, so what you see on the outside, observably. And then homozygous dominant individuals have 2 dominant alleles, which would mean 2 capital letters. Heterozygous individuals have 1 dominant and 1 recessive, which means 1 capital and 1 lowercase letter. And homozygous recessive individuals will have 2 recessive alleles, so that means 2 lowercase letters. So now let's talk about Punnett squares, which basically let scientists break down which alleles and which genes are most likely to move on to new generations. So a simple Punnett square, like what we're going to cover today, will track 1 gene. So you're not going to track more than 1. You're tracking 1 thing at a time. So a female parent that is heterozygous for seed color would then be crossed, for example, with a male parent that's also heterozygous for seed color. And we're going to cover some images in the next few slides that show you a version that you can actually visualize. But, so if we cross a heterozygous female and a heterozygous male, that would mean, if we're using T's, we're again going to have a capital T and a lowercase t for the female, and another capital T and lowercase t for the male. So whenever we run the Punnett square, like we're going to talk about in the next slide, we're able to decide that on average, 3 of those offspring that those 2 heterozygous parents will have yellow seeds for every 1 that has green seeds, which would be a ratio, when we write it, 3-to-1, like in this example here. The phenotypic ratio, or what we're going to see, is going to be 3-to-1, which would mean 75% of them will be yellow, 25% of them will be green, and 2 genes on different chromosomes can be combined into an even larger Punnett square. So we can run these types of tests to test 1, or we can kind of combine them to test multiple. And I will show you an image of actually what Punnett squares look like. We're going to do a few of them together and try to make sure that we understand how they work. But yeah, basically you can have a simple gene that you're tracking, you can track multiple genes, but it kind of gives you a phenotypic ratio that you can write down about average of the offspring that will be passed down. So this is actually a visual example of what we talked about in the last slide. So the same example would be a cross of 2 heterozygotes. So remember, a heterozygote is this type of image. So if we're using F's rather than T's at this slide, the capital F and the lowercase f together represent what we call a heterozygous individual. Now, 2 capital F's is a homozygous dominant, so it's a dominant trait, and little f, or lowercase f, are going to represent a recessive gene and a recessive individual, and a trait that probably won't be passed down. So, in this exact scenario, we are talking about 2 parents, which the parents will always be the outside of the square. So we have a heterozygous, which means we have a capital F and a lowercase. This can be the female. And then we also have a heterozygous male, so capital F, lowercase f. And whenever we cross these, it's almost like multiplication or addition. So we're going to do a capital F here and a capital F here meet in this square. So these 2 capital F's together will end up right here. Now we want to say this capital F and on this square it would be this lowercase f, will come together to be a heterozygous individual right here in this square. So if now we're trying to talk about the capital F here, if we go straight down with the lowercase f over here, once again, we're going to do capital first, always, capital F and then lowercase f. And then if we're going to talk about the last box, we have an f up here that's lowercase, an f down here that's also lowercase that meet in this square, which give us two lowercase f's. So this Punnett square shows a cross of two heterozygotes, which in this case are capital F, lower f, capital F, lowercase f. Only 1 in 4 of the offsprings affected by this disease or this thing are going to be heterozygous recessive. They're only one of the 4 offspring. So if these squares each represent a baby, one of them is going to represent little f, which would be our homozygous recessive. And that's 25% of the first generation, because if we're using 4 boxes, each of them, if we're talking about having 100%, would represent a quarter or 25% of the first generation. And then we have these 2 squares here, the capital and lowercase squares that represent the heterozygous individuals. We have 2 of those offspring who are healthy carriers, which would be the capital F and the lowercase f, which represents, once again, 50% of the generation. And then we have 1 of the 4 offspring up here that's healthy and unaffected, which would be capital F, and that represents another 25%. So that makes up the whole 100 altogether. This one represents 25% of the first generation. So in this case, if we're talking about something that passes down, and we have a healthy and unaffected individual would be dominant, a healthy carrier of that disease will be heterozygous, and someone who's completely affected with the disease will be homozygous recessive. So in this case, we're saying homozygous dominant is going to be right here. The heterozygous are going to be this one, and then the homozygous recessive is down here. So basically, we're saying that 25% of this population, or these 4 children basically, is going to be healthy and unaffected. And we're saying that 50% are going to be healthy carriers of the disease that can potentially pass this down. And then we're going to have fully affected individuals here, which represent the last 25% of the percentage here of, the disease. Okay, so now let's go over what we call an autosomal dominant disorder. So in an autosomal dominant disorder, the parent actually has to carry the homozygous dominant genotype to present the illness. So if you remember, homozygous dominant is exactly what you see here. Every single one of these is a homozygous dominant square. So basically in this case, capital T is the cross shown of 2 parents that are both capital T. So they're both homozygous dominant parents. They would have 100%. So all 4 of these are going to be dominant T offspring. So in this case in particular, a disease that's present in every generation and impacting both males and females is autosomal dominant, and pedigrees can actually help determine inheritance patterns. Okay, so now let's look at autosomal recessive disorder. So it's the exact opposite of what we just saw in autosomal dominant disorder. Basically, we have 2 parents that are both homozygous recessive genotype. So we have little a, and little a on this side. So our parents are both the recessive genotype. So in an autosomal recessive disorder, the parent must carry the homozygous recessive genotype to present the illness. And in this case, that is lowercase a. The cross that's shown here, so the cross meaning this parent and this parent, they are 2 little a or homozygous recessive genotype parents, which means that they have 100% little a or homozygous recessive offspring. So autosomal recessive disorders can actually skip generations and sometimes affect children when neither parent is affected. This would require the parents to be unaffected carriers, and pedigrees, once again, will show family relationships and can help determine the inheritance patterns of diseases. Okay, so now let's talk about what we call a monohybrid. So this is going to represent a kind of weird genetic trait that we'd follow. So earwax is actually a genetic trait. So earwax type is a Mendelian human trait, which means a trait that only follows two possible versions of a gene. So specifically, wet earwax, which would be the dominant disorder here, so the WW that are capital, wet earwax is a dominant allele, and dry earwax, or basically we have the capital and the lowercase on this side, the lowercase would be the dry earwax. It's a recessive allele. So given the parents, in this case, that are capital W, so a dominant parent, and then a heterozygous parent that has a capital W and a lowercase w, genotypes, what is the probability that their child will have the uppercase and lowercase, or the heterozygous genotype? So this is what we call a monohybrid cross, and it shows the possible offspring. So the phenotype wet earwax, which is the dominant disorder up here, is present in 2 of the 4 squares that we have. So if we're doing the cross, remember we go from top to side here. So this square meets on this edge and on this edge. So this would be a capital W, capital W. This square would meet at capital W, capital W. This bottom left is going to meet at capital W, lowercase w, and the bottom right is going to be capital W, lowercase w, right here. So that means that the phenotype for wet earwax, which would be these two guys, they're both dominant, the capital W is present in 2 of the 4 squares. So there's a 50% chance that this phenotype is going to happen with this particular cross. And this is what we call a monohybrid. Okay, so now let's just talk about a few practice problems. So it's just one slide kind of combining everything we've talked about so far. And I once again just kind of want to go over how you go through a Punnett square and come up with the answers. So basically, obviously a square that you draw out will have no answers. So all of these white letters would not be in here. But in this case, we're talking about a cross of 2 heterozygote parents. So remember, a heterozygote would be 1 dominant gene and 1 recessive. So we have a capital B in this case and then a lowercase b. Same thing over here because we have 2 heterozygous parents. So we have a capital B and a lowercase b. Now when we cross these, I always like to start on the upper left. So upper left, we go capital B and capital B. That's where we meet. And then on the upper right, we have a lowercase b and a capital B right here. But remember, your capital is always going to go first. So it's capital and lowercase. And then on the lower left, we have a capital B, a lowercase b. So that's what we have. And then on the lower right, we have a lowercase b and another lowercase b. So in this case, we have 1 dominant, we have 1 recessive, and then we have 2 heterozygotes. So that would be technically 25% of possible offspring would be dominant for the disorder, 25% would be recessive, and then 50% would be heterozygotes for this disorder. So then let's talk again about autosomal dominant. Very easy. We have 2 dominant parents, capital B on both sides, which means that every offspring is going to also be capital B. So we have 100% offspring that are autosomal dominant for this disorder. So they're going to also be dominant. So then we can talk about recessive, same thing, just the opposite. So we have a lowercase b, another lowercase and lowercase b on this side, and all offspring are actually also going to be lowercase b, and they're going to be recessive for this disorder. And then we have what we call a monohybrid. So in this case, we have an autosomal dominant and an autosomal heterozygote actually on this side. So if we do the Punnett square here, we go capital B in this square. We have capital B in this square. In this bottom left, we have capital B and lowercase b. In the bottom right, we have capital B and lowercase b. So in this case, you have 50% that are autosomal dominant, and then 50% that are heterozygote for this disorder. So after all those slides and practice problems, I hope that you sort of understand how Punnett squares work and what they do for us as scientists in determining how our offspring are going to come out with certain genes. So now let's talk about other types of inheritance, because that's not the only way to kind of find out how something is going to be inherited. So in genes with 2 alleles, where a dominant allele masks the recessive, which is always going to happen, the dominant will always mask the recessive, we see the most simple pattern, but gene expression does not always follow that simple pattern. So in these images, I can kind of show you a couple examples. So on the top image, you have incomplete dominance. And then on the bottom image, you have codominance. So these involve more than one allele to encode a functional protein. So in this example, we have a Punnett square actually right here. If you look at the example, it looks almost exactly like what we were looking at before. So we have capital R, which represents a red flower here. And then we have capital W, which represents a white flower here. So if we do our Punnett square, we have RW, we have RW, and once again, we have RW, we have RW. So all of these guys are going to have the option to be pink rather than red or white. So that's incomplete dominance. And then we have something called codominance. So in this case, we have a blue bird, which has two C's is just the bottom example, but your B is kind of what you're paying attention to. So B would be blue, and then in this case, W would be white. So we have a blue bird that has two B's, we have a W bird that has two W's, and then down here we have a blue bird and a W, which would be your white bird. And so this bird actually has a little bit of both. It's blue and white. Like this flower is going to be pink rather than red or white. It's a mix. So you have blue and white on this bird. So basically there's other types of inheritance that aren't so uniform, but they do encode for a functional protein. Okay, so now let's talk about sex determination systems. So in humans, females actually have two X chromosomes, and males have one X and one Y chromosome. So I do have a Punnett square here, which it says at the bottom we want to generate an example. So the egg for the female will always carry an X chromosome, and the sex chromosome in the sperm, therefore, determines if the offspring is male or female. So believe it or not, the male sperm or the male gamete always determines whether the offspring is going to be female or male, because it's the only gamete carrying the potential to be male. So the egg is always going to carry an X, but the sperm can either carry an X or a Y. So mom only gives one of her two X's to baby, and then dad either gives one X or one Y. So if dad decides I'm giving an X, then we have a girl. If dad gives a Y, we have a boy. But the Punnett square kind of breaks down why it's always a 50-50 shot of whether or not you're going to have a boy or girl. So for males, we have X and Y on top. For females, we have X and X. Top left square, you've got two X's. Top right square, you have X and Y. Bottom left square, you have X and X. Bottom right square, you have X and Y. So here on this side, the two X's is going to represent the 50% that could be female. Then the X and Y are going to represent the 50% that can be male. So once again, male or dad always determines the sex of the baby based on whether or not his sperm provides an X chromosome or a Y chromosome. So now let's talk about sex-linked disorders. So some disorders actually carry on based on whether or not the father or the mother is an unaffected carrier or a carrier of a disease, for example, or an abnormal gene. So an X-linked recessive disorder can affect more males than females. So remember, sex-linked inheritance most often follows mothers to sons, which I have an image over here because that is something that I learned when I was in school. So I kind of want to talk about how that happens. So basically a sex-linked inheritance most often follows mother to son. So males only have one X chromosome. To express a recessive disorder, they only need to inherit one X-linked recessive allele. So if we think about this, males, so the sons, only have one X chromosome. So if they're getting an X chromosome from anyone, it's going to be mom. So every time that males get their X chromosome, it comes from mom, which means that if mom has a sex-linked inheritance, some sort of disorder or abnormal gene, that X chromosome is going to go to the son, regardless. So if you look at this image, we kind of break it down by color as well, but we have father here's in blue. He is unaffected, and we're just talking about abnormal genes. So father's unaffected, mother is also unaffected, but she's a carrier of what we call an abnormal gene, which is an orange. So she's half orange and half blue because she carries this abnormal gene. So if they have 4 children, then they have, if we did a Punnett square, we could kind of work this out, but they have the option to have a male child that's completely unaffected because this blue half of mom, so that blue half being her X, and then the blue half of dad end up over here. We also have an unaffected girl, but then we can have an unaffected carrier female. So in this case, she received the gene from her mother, but it affects the son most often because this affected son is going to have that X from mom. So every time that mom has a son, she's going to give her gene here. The reason that this son doesn't have it is because mom still has 50% chance that she's not going to pass that down. So in this case, 50% chance went to this son, and she did not pass the gene down. In this son's case, unfortunately, she did, because 50% of her has the option to pass down an abnormal gene, and this son received her X that had that abnormal gene. So that's how it happens. So most often, disorders will go from mother to son. So I mentioned the word pedigree in a couple of slides previously, and I want to break down what that is. So there are common keys for reading pedigrees. And whenever I think of a pedigree, I think of maybe show dogs or show animals in some sense. If you're in agriculture or anything like that, a pedigree kind of breaks down the family lineage for a dog or maybe a show pig, something like that. So a pedigree depicts family relationships and phenotypes, which you remember, phenotype is what we see on the outside, genotype is what is on the inside. So a pedigree depicts the relationship between family members, and then a phenotype, what is viewed on the outside. Autosomal dominant, recessive, and X-linked disorders can all be identified from a pedigree. Okay, so here are our key takeaways for this chapter. So number 1, DNA codes for proteins. Meiosis allows for alleles to be inherited from both parents. Some alleles exert effects when they are present, which would mean they're dominant, while others are masked, which would mean they're recessive. So if there's ever a dominant around, the recessive will be masked. Number 2 is that a genotype refers to alleles and a phenotype refers to appearance. So a genotype refers to genes, a phenotype refers to what we see on the outside, and a monohybrid cross allows ratios to be written as heterozygote or homozygote, which we covered in the monohybrid cross page. So the last one here is that pedigrees indicate whether a disease is autosomal dominant, recessive, or X-linked. And that is the end of this chapter. Go ahead and move on to the next one. And if you have any questions, please send me an email. Thank you.